Hello, everyone, and welcome to today's webinar, Reverse Engineering the Human Heart with Pluripotent Stem Cells. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by BPS Bioscience. To learn more, visit bpsbioscience.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Aitor Aguirre, Assistant Professor, Biomedical Engineering Chief, Division of Developmental and Stem Cell Biology Institute for Quantitative Health Science and Engineering, Co-Director, MSU Stem Cell Core Facility, Michigan State University. Dr. Aguirre is a stem cell biologist, developmental biologist, and bioengineer who specializes in the heart. He studies basic aspect of human cardiac development, congenital heart disease, and cardiac regeneration. Dr. Aguirre has received numerous awards and published in prestigious journals. Today, he will discuss methods to generate human heart organoids using human pluripotent stem cells. Dr. Aguirre, you may now begin your presentation.
Thank you so much for the introduction, Cassie. And thank you, everyone, for being here today and the organizers for the invitation. So as Cassie said, I'm going to be talking today about how we are trying to reverse engineer the human heart with pluripotent stem cells. So let me get started. So the human heart is a very complicated organ. It's a very powerful pump. And, I like to compare it to an engine. I live in Michigan. As you know, as many of you might know, Michigan used to be the heart of the car industry in the United States and in the world, I would say. And I like to make this comparison of the heart to one of the engines that Henry Ford was making at the start of the 20th century. And so I'm going to use a little bit of a comparison between a pump and an engine here, and I'm going to try to make this kind of analogy. I give this talk to several different types of public. Sometimes it's people that are more into the cardiovascular field. Sometimes it's people that are more just into stem cells. So I have a couple of early slides that are going to be more setting up what's the problem with cardiovascular biology and how we can use stem cells to address that problem. So that said, let's move forward. So the heart, as what would happen with a normal engine, also suffers from wear and tear over the life of over our life, right? And so here I have some pictures and some images representative of what happens with the heart along our lives. On the top left corner, you have there an infarcted heart. You can see the infarct on the wall of the heart, and to the right of it, you have a heart that has hypertrophic cardiomyopathy. So these are parts of problems that constitute cardiovascular disease, which is the main cause of death in the developed world ahead of cancer. And this is a health problem that it's continually increasing and that it's very difficult to manage. And you have a breakdown there of the different types of cardiovascular disease. You can see that the most common one is by far the most, the one with the highest mortality is by far ischemic heart disease. But there's a fair number of other problems associated with cardiovascular health. Another type of problems that we can find with the heart, aside from wear and tear, it's what in engineering terms we would call defective manufacturing. So it's a heart that it's not correctly made. And this is what happens with congenital heart defects, which is another significant source of cardiovascular disease in the developed world. Congenital heart defects are the most common type of birth defect of malformation in humans. And they affect about 1% of all live births during healthy pregnancies. And I have a couple of examples there of congenital heart defects. I'm not going to go a lot into detail into this because this is usually more specific for a cardiovascular audience. But they range from defects on the formation of the walls that separate the different parts of the heart, and defects on the formation of the tubing, basically that joins the heart, which is the arteries and the veins, right? So what kind of tools do we have to address cardiovascular disease, to study cardiovascular disease and to try to identify therapies and new ways to attack this problem? So I would say most of you, if not all, are very familiar with animal models. And I put there 3 pictures of some of my favorite animal models, the mouse, the pig, and the zebrafish. And they have significant advantages. And this has been, I would say, the main driver during the last century, almost, I would say, which is the fact that these models are physiologically relevant. They are evolutionarily conserved, and that's the reason they are useful to study human cardiovascular problems, is because, you know, there's a fair degree of conservation. But they also have significant limitations. One of them is species differences. A mouse is not a person, and their metabolism, for example, is very different. And depending on the organ that you are studying, these differences might be more of a problem, right? Like in the case of the heart, metabolism and how the heart uses energy, and you know, specific parts of the heart, like the epicardial adipose fat. Those are things that are not found in some of these animal models, for example, in the mouse or in the zebrafish. So in those aspects, we are lacking systems that are more appropriate for humans. Another one of the problems that comes with animals is the lack of fine control, and this is just self-evident, right? Like if you have an animal, you know, the physiology of the whole animal will compensate sometimes for problems that you might find in the heart. So that can obscure access to the phenotypes or the features that you want to study. And this is true of all animal models. It's not really specific to cardiovascular disease. And finally, there is, I would say, a recent, you know, rising topic, which is societal pressure against the use of animal models, particularly against the use of some types of animal models. And I think this is a trend that is going to continue, you know, pressing, and it's going to continue getting more popular. So we need to find alternatives to this. And I think, sorry, I continue using my arrow in the keyboard, but I have to press on the screen. This is where human models become useful. So human models, they've been introduced for some years. We started using cancer, transformed cell lines, but in recent years, we have started to use more, you know, relevant, physiologically relevant human cell types, like, you know, pluripotent stem cell-derived tissues. And some of the advantages of these are the fine control that we can exert, because these are in vitro systems, right? So we can control every little component of the medium. We have access continuously to the cells, etc. So we can exert very fine control. But the problem that they have is the lack of physiological relevance, right? And we all know that. I would also like to say that there's also societal pressure to adopt more of these models ongoing in recent years. So the talk is going to focus on our attempts at reverse engineering the human heart so that we can create human hearts in vitro, which is one of the short terms, you know, human heart in vitro human heart models, which is one of the short term goals of our lab. We are also interested in tissue engineering compatible heart parts compatible for transplantation, which is a longer term goal using the same approaches. And eventually, in a future that I might not get to see, we would like to be able to create human hearts de novo, basically in vitro completely. And today's talk, it's going to be focused on this first aim, where we have made a lot of progress in the last 4 years. So to be able to reverse engineer a heart, the first thing that we need to do is deconstruct it. We need to know what is it made of, what is the structure, what are its cell types, right? And I think sometimes there is a persistent thought around people that are not specializing in the heart to think that the heart is relatively simple in terms of cell types. Like most people work with cardiomyocytes 99% of the time. But the heart is actually complex in terms of cell types. It possesses, cardiomyocytes only make up for about half of the heart. And it possesses a series of other cell types. They are described there: fibroblasts, epicardial cells, endothelial cells. There's others. But it also has a very sophisticated structure, right? Like it's a pump, it's a very sophisticated pump. So it has chambers that are separated by valves and a series of connections that are the outflow tract and tubes. And it also is subjected to mechanical forces and blood flow. So it is a physically complex organ, also structurally and anatomically. So we need to recognize first this complexity before we can start trying to separate these parts to reconstruct them in vitro. So I like to show this diagram that shows how a human heart is formed in an embryo, as my first step to explain how we are going to try to reconstruct the human heart. So here you have, starting from left to right, you have the formation of the cardiac crescent, which is the first structure during heart development that will give rise to the heart. And you have the different, I would say, the most important steps there: the cardiac crescent, the formation of the heart tube, early looping, which will give rise to the early structures that eventually will become the different cardiac chambers, septation of the heart, and then after birth, the postnatal heart continues growing significantly during our life, which is also something very important to take into account. So this first structure, the cardiac crescent, it possesses, its form essentially by two populations of stem cells called the first and second heart fields, which will give rise to a lot of the basic scaffolding, we could say, that forms the early heart. But there's other stem cell populations that come and contribute to this structure. One of them is the proepicardial organ, which is you could consider it the third heart field that migrates on top of the structures created by the first and second heart field, and it contributes to proepicardial cells, fibroblasts, blood vessels, etc. Then there is also another population that migrates onto the heart after these three fields have been established, which is the neural crest, which comes from the ectoderm. And these cells, they will migrate through the heart of the embryo, and they will invade and populate the heart a little bit later in development. As you can see, they are about 4.5 weeks after conception in humans. And they will contribute to very important structures too, such as the septum, the outflow tract, the conductance system. And finally, there's also embryonic monocytes circulating in the body of the embryo that will travel to the heart and will get integrated into the heart and become tissue-resident macrophages. And it's been shown in mouse models that these tissue-resident macrophages are very important for the maturation of the heart and basically sculpting that embryonic heart, maturating its blood vessels, stabilizing them, creating parts of the conductance system, etc. So I would say these are the five main cell types, five main heart fields that will contribute to making a heart. But then we also have factors that are exogenous to the heart itself. We have factors like oxygen availability, growth factors produced by neighboring tissues or organs during development. Also, we have the maternal environment, growth factors that are provided by the mother, and nutrients that are provided by the mother through the placenta. And obviously, we have workload. As the embryo grows and becomes more and more independent from the mother, the workload increases significantly, and the heart responds, receives mechanical feedback. A very quick note on self-organization in the stem cells. I want to mention this because a lot of what I'm going to be presenting far down the road is going to be based on self-organization. So I want to make sure that this concept is understood by everyone. So self-organization is a property of stem cells, which means that they exist in a state where they can interpret certain signals, developmental signals, in a way that allows them to create higher organization structures. And this is done through the use of morphogen gradients during development, stable switches. There's other mechanisms. I just want to highlight this aspect. I'm not going to delve too much into it because we could do a whole talk about different self-organization mechanisms and signaling pathways. But my point is that, you start with a single cell fertilized oocyte, and you get a whole human being. So you can, I think that's proof more than sufficient of how powerful self-organization in the stem cells is. And so most of the work that we are doing, it's the work that I'm going to be presenting here today, it's based on the use of pluripotent stem cells. Why do we use pluripotent stem cells? Because they can make all the organs and tissues in the body of the embryo, right? And so this is the perfect setting for recreating organs in vitro if you know the induction steps that will give rise to that organ. So embryonic pluripotent stem cells can be obtained from two sources. They can be embryonic, which was described by James Thompson in 1998, or they can be induced pluripotent stem cells that were described by Takahashi and Yamanaka in 2007. We use both types. We normally use induced pluripotent stem cells for most things because they are easier to justify, but we use both, and the work that you're going to see here today has been performed on both different cell types. And so pluripotent stem cells, as I said before, we use them because they allow us to create unlimited amounts of human tissues in vitro, and so they are the perfect setting for creating human organoids. I'm going to move forward from these. And why are we using organoids? Well, I think organoids are an evolution of the traditional tissue culture and 3D culture approaches that we have been seeing, over the last couple of decades. And by definition of an organoid, and this might differ from the definition of organoid that other people use, but this is the functional definition that I like to use. It makes emphasis on function and structure. For me to consider something an organoid, I like to say it needs to be a complex mixture of cells that recapitulate the structure and function of the organ they imitate to a significant extent. And I like to use this definition to stay clear of things like spheroids or co-cultures of different cell types in 3 dimensions, because sometimes those things are not representative of an organ, or they are only, I would say, tangentially representative of an organ. So that is the definition of organoid that I'm going to be using through this talk. And I mean, I think everyone at this point is familiar with the advantages of using organoid approaches. I like to use this slide from a Nature Reviews paper. But I like to highlight, well, these are some of the advantages, right? But I like to highlight one of the drawbacks of organoids, which is their lack of physiological complexity or physiological relevance. Like, they do have a lot of physiological relevance, but the problem that we have and why they cannot substitute animal models is because they still lack a lot of physiological relevance compared to an animal model. And I think that's a point that we can make a lot of progress, we can improve on. And so there's been significant recent advances on the creation of human heart organoids in recent years. And I'm sure many of you will be familiar with some of these papers. Our work on this started in 2020 with this bioRxiv publication. I think what is interesting about all of these, recent publications is the fact that all of them make a strong emphasis on self-organization, which is what I'm going to be talking about during this talk. So with that introduction done, let's start talking a little bit more about the interesting nitty-bits, which is the results. So as I said before, we are trying to apply a modular engineering approach to building a heart in vitro. So the first thing that we have to do if we want to make a heart, we have to put the basic scaffolding, as I said before, the first and second heart fields. And we can do that, excuse me, we can do that using cardiac mesoderm induction in vitro. This has been described before for a number of years for monolayer and spheroids. These two heart fields will give rise to the ventricle and the atria and parts of the outflow tract, and they can be induced by temporal wind signaling modulation. So basically, if we activate the wind signaling pathway, we will, under certain conditions on pluripotent stem cells, we will get these heart fields arising spontaneously. And I want to highlight that this work was done by one of my first graduate students, Jonathan Lewis Israeli, which was a BME graduate student, and he already graduated. So what Johnny did is he used pluripotent stem cells. He spun them in a 96-well plate, and so he could get embryo bodies, basically, that we then triggered with the wind activation and inhibition typical steps described before to start setting up our protocol. And I would like to say also, before I move forward, that we had a couple of guidelines on how we wanted our heart differentiation organoid protocol to work, and we needed to have scalability That's the reason we use 96 well plates. We wanted to have high efficiency in cardiac differentiation, and we wanted to have very high reproducibility. So these were our three goals when we started this protocol, because we want this to be useful far down the road for pharmacological testing, among other things. So we tested several different cell lines. We could get very reproducible results across embryonic stem cells and iPSCs. And you can see here what the organoids look like, very spherical, fundamentally cardiac, cardiomyocyte-like tissues. And so once we were sure that this part of the protocol was working well, we introduced our next step, which was the addition of a proepicardial organ to this setting. So the proepicardium gives rise to the epicardium. It enables vascularization, contributes to the formation of valvular tissues and some mesenchymal tissues in the heart. And this step could be induced again by temporal modulation of Wnt signaling. And in this case, it's a third step in the protocol shown before, where we activate Wnt signaling at day 7 again. But different from the previous steps shown, this step has to be very well timed. It's a very short exposure, and it only affects the outer layers of the heart. So that way we induce a proepicardial organ without the need to add exogenous cells on the pre-existing organ. And this is what we did here. And you can see here the experimental schematic. And here you can see the proepicardial organ forming and how it spreads out around the organoid as a green layer. And then it penetrates inside of the organoid and gives rise to other tissues. I put two pictures here to show you what these organoids look at day 15. So here you can see the organoids in the 96 well plates. They are these tiny little spheres. They are about 1 to 1.5 millimeters in diameter. Here you can see a close-up. And we decided to do a time course transcriptomic analysis so that we could determine if, the right cell types kind of en masse were being expressed correctly, the right, genomatology processes related to cardiac differentiation and maturation, et cetera, et cetera. And that's basically what we saw. And, we look more in detail into different types of genes that were expressed from the first heart field, second heart field. And some of the stuff that we found was that the complexity of the organoid seemed to be higher than we initially expected. So there were more cell types there than cell types that we had induced. And we suspected this was because of self-organization inside of the organoid. And so basically we saw markers for the conduction system, we saw markers for endocardium, we saw markers for arterial and venous endothelium, for example. And we also saw that these organoids, in terms of complexity, they look closer to fetal gestational hearts from humans than monolayer differentiation cells, which I would say is something that we would be expecting. So we started to look to do some structural analysis to see what were the structures that were found in these organoids. So what we found out is that the organoids would develop a first and second heart field that would actually segregate and would separate and would form sequentially in the organoids. And we also found that we would get atrioventricular specification. And again, we would see these atrioventricular cells segregating and forming a more ventricular and more atrial, structures. We also found that the cell types found in the organoids, there were some cell types present that we didn't induce that were forming spontaneously, like cardiac fibroblasts, as you can see here. And we also saw the position of very rich cardiac fetal extracellular matrix. I'm not showing the data here because we probably wouldn't have time, but that was also a very interesting finding. And we also see that the organoids form nice endothelial cell structures inside of them. And these endothelial cells are embedded into the myocardial muscle. You can see here the endothelial networks, and here you can see a 3D reconstruction. Sorry, I'm saying here because I can see my pointer, but you guys probably cannot see the pointer. In the top row merge, you can see the nice endothelial features forming inside of an organoid. And on the right, you can see a 3D reconstruction done by confocal imaging, where you can see blood vessels in green going through the myocardiac muscle. And we could also see that the organoids form endocardial cells, as you can see here. So we could see these structures that we could see structures that were looking kind of like a spherical inside of the organoids. You can see in the merge, on the top merge there, you can see one of those structures. We were not sure what these structures were, if they were cysts or if they were actually chambers. But what we could see is that there were endocardial cells, positively labeled in endocardial cells that were lining these internal chambers. So we thought this might actually be cardiac chambers. And so we move on to basically verify if these were really chambers. So we contacted one of our collaborators, Dr. Chao Zhou at Washington University. He's an expert in optical coherence tomography, which is a non-invasive longitudinal imaging method that uses visible light. And so it basically uses visible light that can penetrate through the organoid, but it doesn't damage it, so we can do live analysis. And it can penetrate the depth of the organoid, and so basically you can reconstruct the whole structure. And we could basically see very nice chambers forming inside of the organoid. At early stages, which is what is shown here, these organoids have multiple chambers, like usually 5, 4, 6. These chambers are extremely dynamic and they merge. And eventually, after a couple of days, at about day 20, day 25, these chambers usually merge into 2 larger chambers that we will see later on in more detail. On the bottom row, you can see a picture, a bright field microscopy picture of one of our organoids. You can see the organoid there beating very robustly, and you can see the chambers inside too. So this is basically what these organoids look at about day 15, day 15, day 20. Still, they are pretty mature, I would say. We have been continuing to develop these technologies for live non-invasive imaging of organoids, which is a field that is experiencing a lot of growth now, with Dr. Cho at Washington University. And here we have basically developed a microscope that we call the octoscope that can, high content screen of organoids. And here you can see the results. This is an organoid with 2 chambers, and we have this bulb structure in the middle that it's separating the 2 chambers. And when the organoid beats, the chamber closes. And on the right, you can see a conductance velocity map captured at the same time using this methodology. Another aspect that we needed to check with our organoids was the electrophysiological relevance. And this was a little bit challenging because the organoids are pretty big. And when they beat, they move. They beat very strongly, they float, and they move around. But we found a collaborator, Dr. Wen Li, here at MSU, that develops flexible electrodes. And so she basically has electrodes where we can place the organoid, and the organoid sinks inside of the electrode, and the electrode kind of envelops the organoid. And so we could do multi-electrode array analysis using this methodology. And you can see here that the organoids have very nice electrical patterns, and they have actually electrophysiological profiles that are, rather similar to what you would see in a human, right? Saving the distances, but we have curious complexes, P waves, T waves. It looks actually very nice. So I hope that I have convinced you that we have a good organoid model, hard organoid model that models the early heart development to a significant extent. And so once we reached this point, we wanted to start doing some disease modeling to see if our organoids can recapitulate features seen in humans, in human disease. And the system that we chose to study was pregestational diabetes. Pregestational diabetes, as defined as diabetes during the first trimester of pregnancy. And so I'm not talking about gestational diabetes that usually arises later. Now, if a woman has diabetes during the first trimester of pregnancy, which usually means that she's either type 1 or type 2 diabetic person that was diabetic before the pregnancy, the risk of the embryo to suffer cardiovascular congenital heart defects increases very, very significantly. It can go up between 4 to 12 times, up to 4 to 12% of live births, which is a very significant increase. And this is a significant an increase in medical problem. So we wanted to see if we could model this system in our organoids to prove their validity, proof of concept of their use as a medical tool. And so what we did is we recreated the conditions of pregestational diabetes that the embryo says in the uterus in vitro. So we added glucose and insulin in some cases, recapitulating basically the same features that an embryo would see. So I want to highlight that these conditions that we used were very mild conditions. So we are using conditions that are seen in the majority of women. So this is not a level of glucose that is in the extreme range or a level of insulin that's in the extreme range. These are disease-inducing, but in the middle range, because we wanted it to be as relevant as possible, physiologically speaking, for humans. And so what you can see here on the left is normal glycemic heart organoids, NHOs, and PGDHOs stands for pregestational diabetes heart organoids. And so the first feature that we notice is that pregestational diabetes organoids, they grow much faster and larger. And this is interesting because these morphological differences are found in humans too, like macrosomic organs are typical of kids, babies born from pregestational and gestational diabetes mothers. But we also saw that the structure of the organism was affected. And when we did ultrastructural analysis and electrophysiology, what we saw is that at the ultrastructural level, the pregestational diabetes organoids accumulated fat in their cytoplasm. So you can see here the accumulation, like what it says LD, those are lipid droplets. MI stands for mitochondria. We saw a lot of lipid droplets accumulated in pregestational diabetes organoids. And we also saw electrophysiological abnormalities. Sometimes we saw arrhythmias. Not always, but we saw significant arrhythmias occasionally. I'm not sure what is the clinical significance of this, though, because I've spoken with clinicians that tell me that this is clinically irrelevant because these children don't see this fetal, early, pediatric patients don't see arrhythmias. But some of the people have told me that it is actually clinically relevant, and they are also clinicians. So I'm at the crossroads here, but maybe farther in time we can answer this question better. A very interesting aspect that we observe is the development of structural and developmental defects, right? Which is something that I think it's very telling of the abnormalities that this produces in the heart. So we can see, for example, here you can see where it says normal glycemic heart organoids, NHOs, you can see a staining for ventricular and atrial myosins, and you can see that both chambers segregate nicely, and there's you can see chambers inside there. But if you look at the pregestational diabetes-like heart organoids, you can see that the chambers do not segregate; they are actually intermingled to a great extent. And the cardiac muscle there, the MIL7 staining, looks very uncompacted. The muscle looks kind of like spongy, if you want. And we saw that this was very reproducibly found across many organoids. We also did RNA expression for the two conditions in the organoids, and we could see that many critical, transcription factors leading to the formation of the first heart field, second heart field, were dysregulated in time, basically. So they were expressed in very different patterns as they would typically be expressed in a healthy organoid. Now, we can also dissociate the organoids and replate the cells in the organoids for looking at the cardiomyocytes, and that's what we did here. And what we could see is that pregestational diabetes organoids, they develop hypertrophy, which is it's a very well-defined feature in gestational and pregestational diabetes in children. Newborns from pregestational diabetes mothers will have hypertrophy, cardiac hypertrophy. And we could recapitulate that phenotype here with the organoids. We could also recapitulate all the phenotypes that have been observed in mice, such as mitochondrial stress, mitochondrial oxidative stress, and you can see that in the bottom panel. And you can see that the mitochondria, they become like dense spots, which is basically, bloated mitochondria. And we also did perform single cell RNA sequencing to identify widespread developmental lineage alterations. We're interested in seeing how the glucose exposure would actually affect the formation of the different lineages in the heart. And we found very interestingly that pre-gestational diabetes affected cardiomyocytes. It also very significantly affected immature cardiomyocytes. There's a population of cardiomyocytes in our organoids that are still undecided on what they are, and they are still developing, basically. So those progenitor cardiomyocytes were very affected. But I think one of the things that surprised me the most was the effect on epicardial cells. Epicardial cells incredibly expanded in just pre-gestational diabetes-like conditions, and they went from being about 3% of the organoids to be about 25% almost. And I think this is a very relevant finding that hasn't been observed before in mice, because the epicardium gives rise to epicardial fat, which is this protective layer on the outer side of the heart. But it also is a very strong metabolic tissue that regulates access of the heart to fatty acids. And so I think this might metabolically be very important. But anyway, this is an initial observation. We also can see there how the first heart field and the second heart field, as identified by those three markers on each side, it's altered in these pre-gestational diabetic-like organoids. Something that we found that it's new in this system was that pre-gestational diabetes leads to significantly increased endoplasmic reticulum stress. And this endoplasmic reticulum stress in turn leads to abnormal, very long-chain fatty acid synthesis, metabolism in general. So in the upper panel, you can see that the normal organoids, they have very little oxidative stress, but the pre-gestational diabetes organoids, they have higher oxidative stress, but also this oxidative stress, it's localized to the endoplasmic reticulum. When we, and we have more data about this, I'm trying to summarize. And what we see is that this stress in the reticulum leads to a decrease in the synthesis of long-chain fatty acid precursors that are used for the synthesis of very long-chain fatty acids in the peroxisome. And we think this might connect again with lipid metabolism and aspects of metabolic syndrome in the heart. Now, that is all work that it's in progress, and we are gearing up to publish that soon. But I want you to introduce you to another aspect of this talk where we have continued developing the protocol that I initially mentioned. And so after modeling the first heart field and the second heart field and the proepicardial organ, what we are doing now is we are trying to reproduce more of the metabolism and maternal fetal environment conditions. And I like to summarize this. I call it developmental induction. And that's what I'm going to refer to as developmental induction. And it's a combination of factors. It's metabolic factors such as glycolysis versus oxidative phosphorylation. So we provide more relevant media conditions to the organoids, but it's also related to the gradual growth, the use of hormones that are seen during development that are sometimes produced by other organs neighboring the heart or by the mother. And so we designed 4 different medium conditions: a control condition, which is the condition that we have been using to start. Then we develop a maturation media condition, MM, which is a traditional biochemical maturation method that has been, has been reported before. And then we develop 2 additional conditions, what we call enhanced maturation media and enhanced maturation media 2.1, which is a combination of 2 maturation media, in an effort to basically recapitulate more complexity in those developmental induction conditions and see how those affect the development of the organoid. And this is work that is being performed by Brett Bolmer, the very bright BME graduate student in my lab. And so one of the most surprising things that we found with these developmental induction conditions is that they induce or facilitate a patterning of a heart tube-like structure in the organoid. So these organoids stop being such spherical organoids as you saw before, and then they start elongating and developing a much more complex structure. And you can see there a structure of what the heart tube, primitive heart tube looks like. And I want to bring your attention to that retinoic acid gradient there, which starts on the posterior pole where the atrium is forming and the proepicardial organ is migrating. And where there is a high concentration of retinoic acid, and that gradient basically patterns heart along the anterior-posterior axis. And so what we saw in our organoids with these developmental induction conditions, particularly in our best condition, which is that EMM21 that you can see at the bottom there, is that this induces the spontaneous organization of a center that releases retinoic acid. And this in turn enables, patterning along that axis. And we did this using Raman microscopy, with one of our collaborators, thank you, at MSU, that is developing a very sophisticated Raman microscopy scanning microscope that can basically reconstruct the organoids in 3D. And so here you have a picture of what these organoids look like. They certainly look very far away from those spherical organoids that we described in the start. These organoids have two well-distinct chambers. One of them is ventricular-like, another one is atrial-like, and then they have a very nice developed proepicarial organ on top of that atrial chamber, which is, it's very interesting because it's recapitulating this anterior-posterior developmental axis. And then there is an atrioventricular canal that connects these two chambers and that develops some kind of structures that are reminding us of the endocardial cushions, but we still don't have a lot of information about what they are. Yeah, so here you have some more examples of how these developmental inductions leads to the reorganization of the atrial chamber and the proepicarial organ. So basically, here I would call your attention to the EMM21 condition which is the one that it's at the right, where you can see those organoids with that shape reminding a little bit of the embryonic heart. And you can see there the surface of the organoids with that very strong proepicarial organ on top of the atrial chamber. And then you can see the inside of those organoids with very large dilated chambers, right? Much bigger chambers than what we saw before in the more primitive organoids that we were developing initially. And so these organoids develop very nice, again, I'm calling your attention to the EMM21 slide. These organoids develop very nice atrial and ventricular identities, and you can see there I'm staining for atrial and ventricular specific cardiomyocytes, and you can see how they basically situate on two opposite poles of the organoid, again supporting that idea that there's an anterior-posterior axis. And in the bottom layer, you can see the formation of the expression of ALDH1A2, which is the enzyme that synthesizes retinoic acid on the pole opposite to that ventricular chamber, although you cannot see that here because of limitations with the stains, but believe me. And we can also see that there is expression of SOX9. So this is work in progress. We are doing a lot of progress on this area. So I'm giving you here a little bit of a glimpse into what we are doing next. But we also see these SOX9 positive cells that make the valves and the septa, and this has been well described in mice. And they also form in this anterior-posterior pole. And I would like to call your attention to that node of SOX9 positive cells at the tip of the organoid, at the bottom tip of the organoid, which we believe might be starting septation and trying to make a separation between that ventricular chamber into two different chambers. Again, work in progress. We don't really know if that's what's going to happen, but that seems to be what's going to happen. Now, just for you to see that not everything it's perfect, we have also seen that the EMM21 conditions that lead to these more advanced organoids hamper the vascularization of the organoids, and so the vascularization regresses. And you can see here these two animations. You can see how the control organoids have very nice vascularization, but the maturated, developmentally induced organoids have lost a lot of that vascular maturation, vascular integration. We are examining this a little bit more in detail. We believe, we might be able to tune the timing. Maybe we are inducing maturation too fast, and this is producing like a regression of the vascular network. So this, again, is work in progress. Hopefully, we will find out in the next couple of months. We have very nice enhanced biochemical maturity in the organoids after developmental induction. And so here you can see in the EMM21 condition how the number of mitochondria and the shape of the mitochondria, it's improved. We also see a very nice increase in the genes involving oxidative phosphorylation. So you can see those on the right. And you can see the EMM21 condition has very strong induction compared to the other conditions. And we have also enhanced maturation at the electrophysiological level after developmental induction. So we have ventricular, atrial, and nodal-like cells in the EMM21 condition that we do not see in the other maturation conditions. And down here I have some pictures showing calcium waves in the organoid. So this is our control. It fits nicely, but it has some after waves. We have our maturation media condition that has also some has nice contractions, but you can see some waves that are a little bit out of synchronization. Then our EMM1 condition, these organoids beat extremely fast. We don't know why. They are a little bit strange, and you can see that also in the calcium waves on the right, on the left. But here you can see our EMM21 organoids, which is they have very synchronized, very strong beating. With you know, again, it's indicative of maturation. So before I finish with this talk, and I'm approaching the end of my presentation, I want to address the elephant in the room, which is vascularization, you know, blood flow, and yes, this is something that it's very important as cues go for the heart to maturate and develop correctly, and we are missing this step. And this is very challenging because you know, there's a compromise between having high throughput, scalability, etc., and having these organoids connected to a system that can perfuse them and provide correct blood flow. But we are trying to address this problem, and you can see there are several collaborators and people in my lab that are working on this topic, and hopefully we will have some, you know, interesting results in the next couple of years. I would also like to mention that we have successfully, also, I'm not talking about this here today because I don't have space, we have successfully integrated neural crest and tissue resident macrophages in the organoids. You know, and all of these steps are basically modular, so they are plug and play, kind of like Windows. So we can control the complexity of the organoids and we can increase it basically at will, you could say. Anyway, with that, I hope that I have convinced you that we can reverse engineer the human heart to a certain extent, and we can create relevant in vitro models for the study of the human heart for disease modeling. And I would like to acknowledge all the members of the Aguirre lab, our collaborators at MSU, Washington University, Spectrum Health, and other places. And yeah, and that's it for me. If there are any questions, I'd be happy to answer them.
Thank you, Dr. Aguirre, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, I recently read that there are numerous factors that could cause the experiment to include unwanted noise, which could change the accuracy of the findings. What should be taken into account, in your opinion, in order to lessen the amount of noise and inaccurate data used in this procedure?
That's a good question. So I think what you are basically getting to is the problem of reproducibility with organoids. So yes, organoids have certain, I'm going to say bad reputation, but they have a certain reputation for having issues with reproducibility sometimes. And I think it boils down to the fact that as you increase complexity, well, things get more complicated, right? I think the ways to deal with reproducibility are basically hard working into making sure the protocols are as simple and straightforward as possible, and making sure that the quality of the reagents and everything that you use is the best that it's possible, and making sure that people are well trained. But I think an important aspect of this is conceptual. It's making sure that the methodologies that are used are reproducible. So I know that many organoids are made by methodologies that are kind of artistic, right? Like people will take this cell type, this other cell type, and they will put them together. And you know, there's a lot of human factor in there, and that human factor adds to that reproducibility issue. So I think we have to try to get rid of the human factor as much as possible. We have to try to make this as simple and as automated as possible. And that is one of the efforts that we were trying to make here, that eventually I would like to have a robot that can actually do this for me, rather than having a person that has to do it. So I think those are things that will contribute to improving the reproducibility. I hope that answers your question.
Okay, perfect. Our next question here is, as you touched on at the end, for drug discovery applications, you need to utilize high throughput analysis techniques. With the complexity of the organoid models, are they compatible with these approaches, or do novel approaches need to be developed, such as the custom flexible microelectrode arrays your colleague developed?
Yeah, that's a great question, Valerie. There's truth to both sides of the equation, basically. Some methodologies that currently exist can be used, at least in the organoids that we have. But in many cases, we are working at, I would say, almost the limit of what those technologies can offer. So, for example, we have high content imaging microscopes, confocal microscopes that can analyze the organoids and can see through the organoids and take like stacks of pictures, etc. But the organoids are a size at which those microscopes are starting to have trouble, right? So this is compatible with high content approaches, but it's starting to push the boundary of what those high content approaches can do. And I think one of the things that this is going to push forward in the field is the development of new technologies, basically. And we are working on that. You could see that on the optical coherence tomography microscope that we are designing. We need technologies that allow us to look at these organoids in more detail, more straightforward manners, longitudinal analysis, non-invasive, so that we don't have to basically destroy them to analyze what's going on in them. And I think these technologies are going to be key to the advancement of these organoid technologies. They have to go hand in hand. We need the technology with the organoids, and we need the organoids themselves. Yeah.
Okay, thank you. Our next question here, can you touch upon the benefits of using self-organized structures that you mature within the protocol versus 3D printing of tissue models using mature cells?
Yeah, I think that's actually a great question. I hear a lot of 3D printing, 3D bioprinting. I think 3D bioprinting is great, and it's a fantastic approach, and technologically it's a wonder. The problem with 3D printing, I think, at this point in time, maybe in the future this is resolved, it's resolution. So you can macroscopically print something that looks like a heart, right? With a 3D bioprinter, you can put cardiomyocytes inside, and it will look like a heart macroscopically. But if you look at the microscopic level at that 3D printed heart, you will see that the structure is not the structure of a human heart. The cells are not sufficiently densely packed inside of that structure, and sometimes they don't even touch each other. They don't even connect. And so the cellularity is much lower. The structure is not really relevant. So I think 3D bioprinting is currently limited technologically, although it is advancing very fast. So maybe in a couple of years, this will be feasible. But I think right now, the emphasis that we are trying to make is that, there's this saying that says, if it's not broken, don't fix it, right? I think our organs and tissues and cells and stem cells, they know how to make these organs. So why, complicate to make, a very sophisticated methodology to try to reconstruct it with adult tissues when you can actually tell these cells what to do, what to make, and they can make it for you. So in terms of cost, in terms of personal involvement, etc., it's easier to create these tissues with, starting with these approaches that are from simple to more complex, rather than going from a top-down approach. But again, I think both approaches have their values and their strengths and weaknesses, right? So yeah, that's what I think. I hope that answered your question.
Okay, great. Our next question here, asking for more comments about the endothelial cells. As for the vasculature that is observed in the organoids, does it bear markers of cardiac tissue, and are these markers different than the vasculature in other tissues? In pathological models, does the vasculature also seem to have anomalies known of the vasculature present in the human diseased organ?
Okay, so that's two questions. So the first question, we do see some markers that are specific to arterial and venous cells in humans. The problem is that it's kind of hard to tell. We haven't been able to find like nice coronary vasculature markers. If they exist, I would like to know. So if someone knows, please let me know. We see these blood vessels forming, so we don't know if they are more like microvasculature or more like large vessels. But we do see some gene expression markers of arterial versus venous. We do not know if this is coronary vasculature. Again, I would like to be able to know that. So if you know any specific markers for that, I would be very happy to check them out. Regarding the second question, we don't have pathological models of vasculature right now. We haven't been doing that. So I really don't know. I cannot answer specifically if we would see vasculature abnormalities. I imagine we would, but we haven't done it. So it's something I can talk about.
Okay, thank you. Our next question here. If modeling genetic disease, do you start from genetically altered iPS cells or from patients-derived iPS cells? And how difficult is it to obtain cells from patients?
So, yeah, so we can do both. We can start from normal iPSCs, and then we can induce mutations that are, phenocopying patient mutations, or we can get iPSCs from patients. We have both, and we use both. How easy or difficult it is to obtain cells from patients? That is the main issue, right? Like, to obtain cells from patients, you need an IRB. And you need to find patients with your disease, which in terms of genetic mutations might be rare diseases, so there might not be a lot of patients around. So sometimes it's just easier to take the iPSCs and using CRISPR, just edit the affected gene and try to induce the same mutation in a healthy line, basically. This has the added advantage that you also have a healthy control line that is isogenic, which sometimes with patients is difficult to find a good control line. Usually comes from a relative, like a brother or something like that, or parents. So, yeah.
Okay, thank you. And it looks like we have time for one more question today. Do you know if the model of heart organoids is being used to test gene editing therapeutics? Would you expect to encounter issues with penetrance in vitro?
I don't know straight right off the bat if it's being used to test gene editing therapeutics, but it probably is. Since we published this, like more than 80, 100 labs have contacted me to implement these methodologies. So I would guess that some of them might be using gene editing therapeutics. So I wouldn't be surprised. In terms of the advantages and limitations of using this model for that, I think it has a lot of advantages. And this goes in line with that last question, if you would expect to encounter issues with penetrance in vitro. I think the beauty of the organoids is that it gives you more complexity than a single cell layer in vitro, but it's still not as complicated as dealing with a whole animal. So sometimes in animals, you have penetrance issues, right? Because there is a lot of things going on, and you knock out the gene, but there's some compensation, there's robustness in the tissues, organs, etc. In the organoid, we don't have this. We don't have this level of robustness. And issues of compensation can be more, can be controlled better. So if any, we have the opposite problem. We have very good penetrance usually. That's what I would say. So like a phenotype that in mouse, it's maybe more in the gray area. In the organoids, usually it's very clear cut because it's a little bit exaggerated, you could say, because there's not the other tissues and organs around that help, maintain the physiology of that organism. It's not a closed circuit, basically. It's just an isolated part of it. So the organ usually responds very well. And we have done some experiments with this, and it lies with some genetic mutations, and we see very spectacular results from the organoids in terms of structure. The effects on the structure and the cell types, etc., are very interesting.
Okay, thank you so much, Dr. Aguirre. Do you have any final comments for our audience?
No, I just wanted to say thank you very much for attending, and I hope you enjoyed the talk and the information presented.
Thank you again, Dr. Aguirre, for your time today and your important research. We would also like to thank LabRoots and our sponsors, BPS Bioscience, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.